This is a demonstration of running gene set enrichment analysis via the portal. Please note that your titles and colours may differ from those shown here, however the steps of running the analysis will remain the same. Gene set enrichment analysis, or GSEA, identifies the concentration of sets of genes towards the top or bottom of a rank-ordered gene list derived from an expression dataset. The gene sets are held in a database and represent various entities such as experimentally defined collections or genes involved in canonical signaling pathways. The ranking of the gene list is based either on the ability of each gene to discriminate between two sample phenotypes or on the degree of correlation of each gene with that of a query gene. For this example, we will derive a rank-ordered gene list based on correlation of expression with aurora kinase A in the MMRC expression dataset. We will then identify those gene sets in the database which are enriched at the top of that list. We start by navigating to the Data menu and selecting Analysis. We then choose our analysis tool, GSEA, and click Next Step. Now we type in our gene symbol, which for aurora kinase A is AURKA, and then click Select Gene Symbol. As our search returned 2 possible hits, we must choose that which corresponds to our gene of interest and click Select Dataset. Here we choose the dataset in which we wish to perform the analysis and click Select Probe. Commonly, there will be more than one probe for your gene, so you must select that with which you wish to perform the analysis. In this case, there is only one probe, so we simply click Run Analysis. At this point, you may receive a dialog asking whether you wish to trust the site, and you must click Trust Site to continue with the analysis. If you have not run the analysis with that probe in that dataset previously, there will be a wait of several minutes while the analysis completes. When it has finished, you will receive an email informing you. The output of GSEA is somewhat complex, and a more detailed discussion can be found in the documentation within the help section of the portal. We will go through a brief overview here, so we can see the number of gene sets that are significantly enriched at a false discovery rate of less than 25% and at nominal p-values of less than 1 and less than 5%. We click on this enrichment results in HTML link to return a table of the gene sets, detailed on the left, with their size, enrichment scores, and P and Q values on the right. And then we can click on the Details link to look at the details of the enrichment. Here we have a Kolmogorov-Smirnov type plot, which shows how each gene in the gene set falls within the ranked list of genes. Below the plot is a table of the various genes in the gene set. There are annotation links on the left, and then on the right is detailed whether the gene contributes to the enrichment. Below the table is a heat map with samples organised in columns and genes of the gene set organised in rows. And here we can see high coordinated expression of members of the gene set in various samples. GSEA can be launched in other manners within the portal. So, for example, we can enter our gene of interest in the search box, click Search, Analysis and GSEA, which launches us directly into the wizard. Alternatively, we can click Data and Browse to look at the data sets. Select Data Set of Interest, Analysis and GSEA to launch us into the wizard. This has been a demonstration of running GSEA via the portal. Please note that your titles and colours may differ from those shown here. However, the steps of running the analysis will remain the same.